So today we're looking at measuring abiotic, which is another word for non-living factors in the environment. So some abiotic factors include temperature, light intensity, moisture content of the soil, pH of the environment, which can include pH of the soil, rainfall, oxygen concentration of the air, the soil and the water, etc. So to measure light intensity, you use something called a light meter, which has a sensor on the top, and light intensity is measured in lux. So what you would do is you would put the light meter in the area that you would like to measure, and you would face it towards the source of light. So if you're measuring the light intensity of sunlight in a wood or under a tree, you would face it towards the sunlight that's coming through the leaves or the gaps in the leaves. Then you would read the scale, repeat probably about 3 times, and then move to a different area. For soil pH, you would use a pH probe. Literally just put the end of the pH probe into the soil so it just covers the tip. You record the pH off the scale. You could probably repeat that 3 times and then move to a different area. Before you start on your next area, you'll have to wipe the probe down to make sure there's no residue left on it. Okay, then you can also measure soil moisture. So you would use a soil moisture sensor or a soil moisture probe. And again, you would put it into the tip of it into the soil, record the moisture, repeat a few times, several times, and then you would move to a different area. To measure air temperature, you would usually use a maximum min thermometer, max min thermometer, which has a temperature recall function, so it allows you to see what the maximum temperatures have been throughout. Then you can look at soil temperature, so you'd use a soil temperature probe. You'd put the probe in the soil, covering only the tip, record the soil temperature, and then repeat several times and move to a different area. Rainfall, you'd use a rain gauge. This just collects the rainfall and you can measure the volume. You leave it for a set amount of time. So if you wanted to find the rainfall in a day, you'd leave it for a day. If you want to find it over a month, you'd leave it for a month, etc. You've got dissolved oxygen concentration, which you can use to find the oxygen concentration in water such as rivers, lakes and ponds. You place the oxygen probe into the liquid, record the concentration of oxygen, repeat several times and then move to a different area. And that's it for abiotic factors or non-living factors.